Hi guys, welcome to your first official day of lecture and of class. What I want to do today is start at page 15 in your binder. So we're going to go from page 15 to 26 in 25 minutes or less. I'm going to just discuss some topics that you already are really familiar with. I normally start the year with a scientific method and a really great design your own experiment where we look at animal behavior. Since we're not in the classroom though, I'm going to kind of just really skim the surface of this for completion's sake, and then we are going to get into biochemistry later on this week, and then we will come back to the scientific method once we start getting students back on campus and we can really start looking at designing some experiments using some of the really great supplies that are in the classroom. It's just not something that you can do from home. And so the scientific method you probably already saw in all of your other science classes. And so this is going to be pretty easy. If you're on page 15, just follow along. I'm going to have you write down a couple of things. And then I'm going to just kind of breeze through chapter 1 for this class. It's just a broad overview of what it takes to design a good experiment and what the 5 themes of this class are. So the scientific method, it's a way to solve problems. And you use it every day. We all use it every day. Some questions that you might answer each day are: where are my shoes? What should I have for lunch? What class do I have next? Did I do my homework for that class? In this class, we're going to talk about cancer a lot. So what is the cure for cancer should come up a lot in this class. And then, some people might want to know which deodorant works the longest, and they might conduct their own little experiments for that. So we use the scientific method to answer a lot of questions in our everyday life. So on to page 16. The steps of the scientific method have to be followed in order. So the first thing that you're going to do is come up with a question, or you're going to have a problem that you want to solve. You'll need to research information. You'll need to form a hypothesis. And in this class, you're going to do hypotheses different than how you've ever done it before. You'll design an experiment, or you'll perform an experiment that's already designed for you using equipment and materials from the classroom. You'll make observations and you'll collect data. And then you will form a conclusion based on the data and observations that you collected. All right, so scientists are going to develop a question about a problem. They need to be very specific in defining what they are trying to explain or solve. And it's got to be an actual question or problem that we can solve in class. So one of the problems that we would not be able to solve in class has to do with like spirituality and stuff like that. Like, is there a God? That's probably not something that we're going to be able to do during class. So it's got to be something that we can actually solve, a problem we can solve or a question we can actually answer. So after you've come up with your problem or your question, you're going to need to research information. And we research information usually using the internet. So what I'd like for you to write down next to this is, because we're not going to do books, magazines, and all of that stuff. We're not actually going to go to the library to do this, because why would we when we have the internet? What I want you to write down is the only acceptable sources for your information are .org, .gov, .edu. .org, .gov, .edu. If it's .com, we really just don't trust it. Now, every once in a while, we'll have a student say, Dr. Holbrook, look at this .com source. I mean, come on, this is definitely legit. And I'll be like, all right. So if you're going to use a .com source, at least pass it by me first. All right, so we research information, and then you develop your hypothesis based on that information. What you are normally used to doing is the if-then statement. So I bet you guys are used to making an educated guess and stating, if blank, then blank. If I do this, then this will happen. And that's great, and that's a great way to teach hypotheses and to teach the scientific method for middle school and for 9th and 10th grade. But once we get into 11th grade, we do big bad statistics. And with statistics, you cannot make an educated guess. You cannot make an if-then statement. Every once in a while, I'll let you do it when we're not using statistics. But when we're using statistics, you are going to follow this method of forming hypotheses right here. So You're not going to be making predictions on what you think will happen unless there is an actual rule, like a genetics rule. And we're going to go over that when we get to genetics. So just know if there's not like a hard, fast rule, then your hypotheses are going to look like this. Null hypothesis and alternative. You're going to have both. Your null hypothesis states there will be no difference between my control and my treatments. So I would write that in. There will be no difference between the control and the treatment groups. The alternative hypothesis is there will be a difference between the control and the treatment groups. So let me give you an example. Let's say I have brine shrimp, and the question I want to answer is, what type of water will make brine shrimp eggs hatch best? My null hypothesis is there will be no difference between the water treatments. So if I have just regular tap water, 1% salt and 2% salt, my null hypothesis is that I will see no significant difference between the hatching rates in those 3 petri dishes. My alternative hypothesis is there will be a difference in hatching rate. And this is something that I want to make sure everyone understands, and so I will be visiting this topic in our next Zoom, what null hypothesis and alternative hypothesis is. At the end of your experiment, when you do your statistics, one of these will be true. Either there was no significant difference between your control and your treatment groups, or there is a significant difference between your control and your treatment groups, and then you're going to state what you think that difference is. All right. You're going to then create and conduct an experiment to test your hypothesis. And so on our second meeting, you're going to do an experiment. It's a virtual experiment where you're going to look at different environmental factors and how they affect the chirping rate of crickets. So you'll do a little bit of research, kind of figure out, okay, temperature, humidity, other crickets that are around. What do I think is actually going to happen with all those different variables? And then you're going to you're going to do 5 different experiments on the cricket. And so you have materials and you have a list of your procedure, step-by-step directions. For my class, when we get back into the classroom, I want you to have this handy because I'm going to expect you guys to already know when you write a formal report for me, your material list is in bullet points. Your procedure is numbered. So put a star by that. You're going to have to refer back to this lecture when we do our actual in-class experiments and write your reports. Let's talk about variables. So a variable is anything that changes or could change an experiment, and there are 3 types of variables in an experiment. The independent, the dependent, and the constant. And you guys already know this. Especially if you took cell bio this summer, you know this because I know what all 3 teachers did, and we all did something on variables. The independent variable is what the scientist is manipulating. The dependent variable is what the scientist is measuring, right? It's the part of the experiment that changes as a result of some other action is measured. And then the constant is a variable that does not change. So let's take my brine shrimp example. I make 3 petri dishes. I'll just remind you how I'm setting this up. 1 petri dish has tap water, 1 petri dish has 1% salt water, 1 petri dish has 2% salt water. I put brine shrimp eggs in all 3, and I want to see, is there a difference between the control and the amount of salt that I put? What am I manipulating? Well, I am manipulating the amount of salt. So the independent variable would be amount of salt. What am I measuring at the end? I'm measuring the hatching success rate among the 3 different petri dishes. So the dependent variable would be hatching rate. What am I keeping constant? Well, some things I'd like to keep constant is amount of light, temperature, and water quality, right? I want to get from the same source of water, right, that I'm mixing the salt in. If I use tap water in the control, I better use tap water in all the other ones. And the same, if I use distilled water in the control, I better use distilled water in the other ones to mix with the salt. So those are our 3 different variables. We keep some variables like temperature and light constant because we want to make sure that there are no other variables that can actually be having the effect on the dependent variable and is accounting for any differences that we see, because the independent variable is the only thing that we want directly linked to what we're measuring. All right, you need to really think logically when you're investigating a question or a problem and break things down into many steps that make sense. So your procedure needs to make sense. I need to be able to read it, and I need to be able to reproduce what you did in the lab when you are writing a procedure. I'm turning my page right now. Where are we? All right, observations. So when you can take qualitative data, or you can take quantitative data. So qualitative data is going to be color. If you're looking at bubbling, how vigorous something bubbles. Qualitative data isn't quite as reliable as quantitative. And so what you're going to do is record qualitative and quantitative depending on what experiment you're going to do. And that is basically gathering information on what happened. And so the most important thing about observations is that they need to be organized. And in this class, I know it says charts, graphs, written work, all of that stuff. Data, your data section is going to be a table every single time. It's got to be an organized table every single time. All right. Your analysis is then taking your data table and making a graph out of it. You will always put your graph under the analysis section of your report. And remember, don't get freaked out this week. We're not actually writing your formal report yet, but we do have to start the year with scientific method because it's an easy thing to start with. I don't want to start talking about biochemistry on the first day because then everyone's it'll turn you off. Scientific method's always something like, yeah, okay, that's familiar. I can do this. But just know you're not writing your formal report yet, but you need to refer back to this because I'm not going to do this lecture again later. Under analysis is where your graph goes. And your graph is based off of your data table, right? So which graph do you use? You have two choices. You have a line graph or you have a bar graph. And you can't just pick one. You have to pick it. It has to make sense. Anytime you have time or temperature, you're going to use a line graph. Anytime you're comparing different groups, you're going to use a bar graph. Anytime you're doing a formal report and you're just really not sure, you need to ask me. But I am telling you, every single year on the AP exam, this question comes up every year without fail. It gives you a data table and it says, all right, which graph do I use? And it'll give you 4 or 5 different graphs. Never pick the pie graph. We will never use a pie graph in this class. If you have time and temperature, use your line graph. If you're comparing different groups, use your bar graph. Those are really the only 2 options that we should use. Under your analysis section, your statistics calculations will go here. We are doing statistics in this class. Yes, so if you're taking AP Stats, you're in luck. You're going to do, anyone taking AP Stats always really understands, and then I rely on the AP Stats students to help with other students as well. Under conclusion usually just is what did you find the answer to the question. So I want you to kind of draw like a little circle around conclusion and this first bullet point. That is really the only thing that goes with conclusion. And I probably should have just reformatted this slide, but this is my second time recording. I'm not about to re-record again because I got to move on. So under conclusion, and this will just tell me like who actually paid attention. Under conclusion is the first bullet point. Then I need you to write the word discussion. Discussion. Right here where you see my mouse. Discussion is where all of these other bullet points go under. Your conclusion and discussion are kind of together, but when you're making a formal report for me, under conclusion, I want 1 sentence. What did you find the answer to be? And then under discussion, I want you to answer all the rest of these. What were the sources of error? What could have influenced your results? What changes would you make next time? What other questions could you investigate? And then was there anything wonky with your statistical analysis? Okay. Awesome. Once you complete your experiment, you need to make sure that you redo your experiment because it needs to be redone over and over. I like to tell students to do it 3 times. 3 times and then we're good. If you do it 1 time, you really can't make a good conclusion. So all this slide is telling you is repeat your experiment at least 3 times for verification. And then the last thing that you will do is you'll share your findings. You'll share your findings in the form of a formal report, or this week in this class, your experiment is designed for you. You're going to share your findings with a little mini report. Cricket experiment will be a mini report, and you'll turn that back into me. Okay? And then when you're writing a formal report, this is the format we're going to follow. These are the bolded sections of your formal report. It will be in this order exactly. You need to do MLA formatting for your references. And then remember, .gov.org.edu. If it's a .com, it needs to be approved through me first. And so that's what a formal report is going to look like. I will tell you guys right when we get the notification that we are going back into the classroom, even if it's just half of my students, I am just going to cancel everything I originally had planned. We are going to design an experiment and we're going to do this because it's so important to do the science, not do virtual labs. Virtual labs I like, they have their place, but right when we get back into the classroom, students in class are going to partner with students that are out of class, and you guys are going to design an experiment and you're going to collect data and you are going to write a beautiful formal report. I just can't wait until we get to go back. Okay, we are moving on to page 23. I still have some time left and I want to go over chapter 1. So this week I am just really kind of breezing through chapter 1 and chapter 2 because chapter 1 is just telling you what are the 5 themes of this class and a little bit more information on what makes a good experiment. So I'm just going to breeze through this because you don't need to know everything about all of these themes yet because that's what we're doing in this class. I just want you to know what are we going to be studying. So what I want you to know when I get done with page, let's see, we're going all the way to through page 26. So I have 3 pages left in about 8 minutes or less. What are the 5, what makes something living? What are the 5 themes of this class and what makes a good experiment? And so our class, the overarching theme is evolution. Okay, the overarching theme is evolution. So I want you to know that right off the bat, that evolution is the driving force behind everything we do in this class. I'll bring it up frequently. Evolution is the change in the genetic makeup of populations over time. And so we're going to spend a lot of time on that. But we need to study some things first, which are biochemistry, cells, DNA, mitosis, cell division, all of that stuff. And we're going to study a lot of diseases along the way because I like to throw diseases in there. Studying evolution and studying diseases actually can go hand in hand. I'm going to have some activities for that too. So biologists are going to study cells, tissues, plants, insects, bacteria, diseases, and genetics. And we are going to study all of those things. I'm most looking forward to our bee, our ecology unit where we study bees. We're going to do some bacteria culturing. We're going to grow some plants in the greenhouse. And we're going to definitely look at a lot of diseases and we're going to do a lot of genetic stuff. So a lot of stuff this year. And so before we begin though, I want to make sure you understand what is considered living. And so I know everyone got this in cell biology, but just to remind you, what makes something living is this list right here. Growth and development in order, homeostasis, adaptations, evolution, all living things evolve. Response to the environment, reproduction, metabolic processes, and all living things possess a lifespan. What I have over here on the right side is all the things that are things that are considered, not all the things, but things that are considered non-living that some scientists will say, well, they're kind of living. And I agree with that. I think viruses are kind of like a bridge between the living and the non-living, which kind of sucks because they're so harmful. But this one down here is Ebola. Ebola has this really specific shape right here that you can always recognize when you see it. Right here, this crazy thing is called a bacteriophage. It's a virus that actually infects bacteria. Here's a bacteriophage right here. And so we'll talk about viruses in this class. I love talking about viruses, especially in the current climate. So I'm going to talk about viruses and I'm going to talk about the COVID vaccine, especially because I'm in the trial to get the COVID vaccine. So, which is kind of scary, but I'll talk to you guys about that later. But what makes a virus non-living is they do some of these things, but one thing they don't have is metabolic processes. So viruses aren't, they don't eat. And they don't do cellular respiration. They don't make ATP. They don't use ATP. So viruses are considered non-living. Okay. Our first theme, and I'm skipping over slides that you don't actually have in your notes. Our first theme is that new properties emerge at successive levels of biological organization. And so life can be studied at different levels, and we're going to do that in this class. We're going to start at the micro level, biochemistry, Then we're going to talk about cells. Then we're going to talk about tissues and how cells divide to make tissues. Then we're going to talk about evolution, and then we're going to end the year with ecology. And so this is our hierarchy of life. You can start over here. I like to start over on this side and I go to the left, smallest to largest. So atoms make up organelles, make up cells, cells make up tissues. Tissues make up organs and organ systems. Organs make up organisms. Organisms make up populations. Populations make up communities. Communities make up ecosystems, and ecosystems make up the biosphere. So we are going to start over here. We're going to talk about water and atoms and stuff tomorrow, or the next time I see you. And then we're going to move all the way through and end the year with ecology. Theme 2 is life's processes involve the expression and transmission of genetic information. And so we're going to spend a lot of time talking about chromosomes, DNA, genes, genetic expression, regulation of genes. So all I want you to know is the theme and what goes under it. So I'm going to kind of move fast through these because I just really want you to know the theme because I'm not going to get into all the content under the themes yet. And so this is a picture of fluorescent microscope of mitosis. So we're going to go through mitosis. We're going to talk about the structure of DNA and the function of DNA. And one of my favorite things to talk about is embryo development. We're going to talk a lot about embryo development and hopefully do some research on zebrafish embryos. Theme 3 is that life requires the transfer and transformation of energy. And so we're going to talk about how energy gets incorporated into living systems from the sunlight. We're going to talk about photosynthesis. We're going to talk about cellular respiration. We're going to talk about energy transfer between organisms. So just energy is theme 3. We're going to talk about, like I said, energy exchange between organisms and the environment, how energy flows through ecosystems. Photosynthesis and cellular respiration go under this. So hopefully it doesn't feel like I'm going too fast. It's just that you don't have to have any of this stuff memorized. What I really need you to have memorized are what I said before, what makes something living, and then what are the 5 themes. Organisms interact with other organisms and the physical environment. So we're going to talk about relationships, symbiotic relationships between organisms, and interactions of organisms in their environment. We're talking about trees taking up water from the soil. We're going to talk about how bacteria fix nitrogen in the soil in the nitrogen cycle and stuff like that. We're going to talk about cycling of nutrients and the flow of energy and ecosystem dynamics. And you'll see this picture again. This just shows the flow of energy through an ecosystem. And then we're going to talk about evolution. Evolution unifies biology at different scales of size throughout the history of life on Earth. We're going to talk about a lot of different species in the history of life on Earth. It says 1.8 species have been identified and named to date, and thousands more are identified each year. And that the estimates of the total number of species that actually exist range from 10 million to over 100 million. And then we're going to look at things like this. This is a phylogenetic tree right here. And so those are all the themes. I just want to end with talking a little bit more about experiments and designing experiments. A controlled experiment is going to compare an experimental group with a control group. So remember in my brine shrimp example, the control group was just plain water and the experimental groups was the different amounts of salt. A controlled experiment means that the control group is used to cancel the effect of unwanted variables. It does not mean that all unwanted variables are kept constant. So when you write your discussion, we'll talk a lot about what unwanted effects may have not been kept constant. But just a control means that there's no treatment. There's limitations to science. In science, if you're going to do an experiment, it has to be repeatable. We can't talk about supernatural explanations, which are outside the bounds of science. In the context of science, a theory is something that's broad. It's broader than a hypothesis. It can lead to new testable hypotheses, and it can be supported by a large body of evidence in comparison to a hypothesis. So evolution would be an example of a theory. And the most complex experiments are going to involve modeling, laboratory experiments, and then we're going to do things in the field, like outside. And then we can model things in this class. So we can make diagrams, three-dimensional objects, computer programs. We're going to look at some computer programs, and we're also going to look at some math when it comes to seeing if a population is evolving. So I just want you to know that models are a representation of natural phenomena and that we're going to be using some of those in this class. This is a model of how the heart works and how blood flows through the heart. And then in this class, we're going to work in teams. So the culture of science is to work in teams and to communicate your results with each other and to come up with ideas with each other. So that's the culture. And here's a cool picture of two people working together. So you're going to be working together. Even if you're at home, you're going to have a group that you work with. And then lastly, the combination of science and technology will have a dramatic effect on society. And so we're going to talk a lot about ethical issues with technology. There's a lot of politics, economics, and cultural values that kind of can make things a little bit complicated, but hopefully we can approach that with sensitivity, but it is something we are going to approach as some ethical issues in this class and discuss them as a class like adults or soon to be adults. And so that's really all I have for you. I hope I didn't go too fast through that. I feel like most of the stuff I talked about you really already knew, but for completion's sake, I wanted to make sure I just kind of glossed over chapter 1 and the scientific method. For your next lecture, we will do chapter 2, and that should take definitely less than 25 minutes. I'm sorry I went over my time a little bit this time, but hopefully you got a good overview of what we're going to be doing in this class and what experiment design and the scientific method is going to entail. I will talk to you guys later.